So I want to welcome everyone today. I'm Kelly Eversole of Eversole Associates and one of the founders of the Women in Genomics group. We were actually founded in 1998, a long time ago, at the Plant and Animal Genome Conference, and it was meant to be an annual gathering of women to network and of both the international and people at different career levels and give people enhanced opportunities to support each other. And it's been going on very well. And in the past couple of years, we've kind of expanded to do something outside of the Plant and Animal Genome Conference and have had one other webinar where we brought a woman in to have a conversation with us. And today we're moving forward with that as well and hope to do even more. I am joined today by Deb Hammernick, Laura Mathieu, Charlotte Rambla, and our guest, Pamela Ronald. We are the leadership of the steering committee, organizing committee of the Women in Genomics, and we're very pleased today to welcome Pamela Ronald as our guest. And I do want to just to let you know that we do have a few events coming up in the new year. There will be a workshop at the Plant and Animal Genome Conference and our dinner. And at the workshop, we will have a number of distinguished female scientists and some of our male allies who have helped women. And then we'll also, as part of that workshop, have an opportunity for speed networking, something that hopefully you'll leave the workshop with new contacts. In addition, we'll have our quarterly call, which is just a get together of everyone at the end of January. And also we'll be doing a joint breakfast, Women in Networking breakfast with the LGBT Ag group. And that will be in Phoenix in April. So be sure and sign up for the Women in Genomics mailing list so you can stay abreast of everything. So I want to welcome everyone today. And at this point, I am going to be turning it over to Charlotte Rambla, who will take over from here. So thank you, Charlotte, for taking over, and thanks everyone for being here today.
Thank you, Kelly. So good morning, good evening, wherever you are. And welcome to this Women in Genomics webinar. And thank you so much for joining us today here. So first, I would like to remind you, everyone, that please use the Q&A box to enter the question here below. And we don't have the chat function enabled for this webinar, so please use the Q&A box to ask questions. And also please keep an eye on the chat because we'll be sharing very useful information and important resources there, so please keep an eye on it. And lastly, today's conversation will be recorded and archived for future references and on the Women in Genomics website, and that is womeningenomics.org. That's it. So yeah, I'm gonna hand it over to Laura, and yeah, that's it.
Yeah, so today in this webinar, we are pleased to talk with Dr. Pamela Ronald, so professor in the Department of Plant Pathology and the Genome Center at the University of California, Davis in the US. So Pam is a universally recognized leader in plant genetic, a food security pioneer, and an enthusiastic advocate for sustainable agriculture. So as a professor at the University of California, Davis, she has dedicated her career to understanding how plants respond to disease and environmental stress. And her pioneer research led to the isolation of the sub1 gene in rice, which confers tolerance to up to 2 weeks of complete submergence. And this discovery facilitated the development of high-yielding sub-1 rice varieties, which have been adopted by millions of farmers in the South and Southeast Asia, which dramatically improve food security in flood-prone regions. She's also known for isolating the XA21 gene, which improve broad-spectrum resistance to bacterial blight in rice. And beyond her laboratory work, Pam is a leading public advocate for using modern genetic tools, so including biotechnology and gene editing to address global food challenges. She has co-authored the book Tomorrow Table, advocating for a science-based approach to agriculture that integrates both the genetic innovation and the sustainable practices. And her effort in research and communication has earned her numerous accolades, so including the Wolf Prize in Agriculture. So thank you very much, Pamela, to be there and to accept discussing with us today. So we have a long list of questions for you. So let's get started.
Yeah, let's get started. So I'm very excited of having you here, and I can't wait to ask you all these questions.
Well, thank you both. Thanks for the nice invitation, and many thanks to Kelly Eversole and the whole team for putting this together. It's quite an honor.
Okay, after like your biography, that was amazing. I'm gonna start asking the question. So just to give like a bit of background about like and to get the audience to get to know you a little more after all this very knowledgeable biography, I would like you to tell us yeah, where you grew up and your early interest and the path that led you to science and to who you are now.
Okay, thank you. Well, I grew up in the Bay Area in a city called San Mateo, between San Francisco and Palo Alto. And then my parents, before I was born, they built a tiny little 500 square foot cabin in the mountains, in the Sierra Nevada mountains. And we spent all our winters and summers up there. And I loved backpacking very young with my brothers. And we'd go out for days and we would tell my mother where to pick us up, and she would be there. No cell phones, but it always seemed to work out. And I just loved being up in the mountains. And once on one of those backpack trips, it was a very hot day, I remember, when we got up to a saddle, and there was a man and a woman who looked very relaxed, eating their sandwiches with a big book on their knees. And I'm like, well, what are you doing? And they said they were identifying plants. And they worked, I think, for state organizations. And I was pretty young, probably about 14, maybe 15. It was the first time I think I really thought about that I was going to grow up and have a career. And it just hit me, well, I want to do that. I want to be in the mountains identifying flowers. So that's what got me very interested in plant biology.
And so can you tell who have been the most influential mentor of or role model throughout your career? Like, and what kind of lesson did you take from them?
Okay, well, in college, I had wonderful role models. Bert Bram took us out I took a botany class in college and identifying flowers in the Pacific Northwest where I had moved to college. I worked with one of the first plant biologists in the country to have a job. Her name was Helen Stafford, and she recently was awarded a prize from the American Society of Plant Biology to recognize her contributions. And we were all in awe of her because we understood that when she was coming up through science, there were not many opportunities, and Reed College was one of the few places that would offer her a position. And then, I mean, really at every step, I had just tremendous mentors. After college, I tried to find a job at Stanford, and eventually I ended up working in Virginia Walbot's laboratory as a lab assistant to Judy Callis. And they were both very supportive, very fun. At the time, the lab had 16 women and just one man. And so we were always laughing and talking, really loved it. And I have to say, that's when I totally fell in love with research and got a lot of support from Jeannie Walbot to pursue a master's degree and eventually a PhD. When I was at Stanford, there was the Carnegie Institute of Plant Biology at Stanford was a wonderful place on campus. Unfortunately, it's been dismantled. Never would always go. I heard Brian Staskowitz talk about plant microbe interactions, and I was very interested in his work. So I ended up applying to be a graduate student in his lab. And then for the postdoc, I wanted to contribute to global food security. So I had been working in pepper and tomato and, working on salad ingredients, and I wanted to work on a staple food crop. So I moved to Steve's lab to work on rice, and I had a lot of support from Jan Leach to help set up the rice-Xanthomonas interaction, and that really launched me on the rest of my career.
That's awesome. Like, I love that. And thank you so much for sharing all this. And now we're gonna go. So like we know about what you did, and everyone knows what you did and your incredible research and science. So we were wondering through your career, which of your project or discoveries has been the most engaging or interesting for you, and we really want to know why as well. Thank you.
Oh. Well, okay. So the first big discovery with my team, and I always have to say, by the time I was a professor, I had incredible postdocs leading the project. And at the time, that was Wenyuan Song, who's now at the University. I had mapped XA21 when I was at Steve Tanksley's lab, and everybody working to try to isolate the gene, and we found a set of genes that were very interesting. They were receptor kinases at the locus time. The only receptor kinase that had been characterized was the SRK, the SERK receptor. And we put all the genes, there was like 6 of them, we put them all into plants to see if they conferred resistance, inoculated them all. And I remember Wenyuan Song and Guoliang Wang saying, you have to come to the growth chamber because we think we have the gene. And I came to the growth chamber, and there was a lot of plants, because they inoculated all of them at once. It was a huge experiment. So there were a lot of plants that had long streaks of bacterial infection. And there was a set of plants that looked like they had not been inoculated. It looked like, just there had been a massive haircut with green snippets everywhere. And this one set, I had to ask them, are you sure you inoculated it? Because they were so resistant. And that's when we were able to confirm we had isolated the gene XA21. It was a really exciting time because there was a lot of interest in that gene. There had been a really short period of about 3 years where scientists in many parts of the world were isolating resistance genes. And maybe just to go back a little bit, breeders had been breeding for resistance for about 100 years. A scientist in England had been able to show Biffin that you can cross a resistant line with a susceptible line, and you can get resistance, and that was heritable. So breeders have been introducing these genes for so long, but no one knew what the genes were. So in a period of really about 3 or 4 years, we isolated many genes, many types of genes. NBS-LRR genes were a big class of genes isolated in Australia and Berkeley. And the gene we isolated was in a separate group. It was a receptor kinase, so it had a receptor domain and a kinase domain. So we could imagine that the receptor domain would sense the bacteria, and the kinase domain would signal. So that was certainly a very exciting point early in my career. And there have been other moments as well, for sure.
Yeah, so I think this kind of project you will do exactly the same if you have to do it again. But is there anything from your career that you wish you will do it differently if you go back?
Oh, well, yeah, we had a big problem about 10 years ago now. So after we'd isolated XA21, we shifted to the bacterial side, and we were trying to find the molecule that triggered the resistance. So typically with the receptor kinase, the receptor domain will recognize a product from the bacteria. Well, I had a team working on that, and we thought we had isolated it, and we published it, and it was wrong. And we had to retract the paper, and that was really, really very difficult in many, many ways. And I had people in the lab were thinking of leaving, and I said, I understand if you leave, I'll write you strong letters. But really what meant so much to me is everybody stayed. And not only did they stay, they redoubled their efforts trying to isolate this gene. And then Rory Pruitt in my lab, make a long story, he isolated the gene from the bacteria that we named RAXX, which could activate XA21 mediated immunity. So that was another very, very exciting moment. And then we published the gene. So I wish I had not had to go through the whole retraction process. It was very, very difficult. Of course, there were things we could have done differently at that time.
I mean, that's life. I mean, we learn from our mistakes.
Yeah, well, we learned a lot, but as scientists, we don't expect to publish a paper that's wrong.
Yeah, I know, but I mean, we're human, so I mean, that's life. It's okay. After like you've done an amazing, like your career is amazing, so it's just like a small thing that happened in your life. And through this, I would like to since like all your wisdom and all your knowledge and all your insight, I would like to know like if you could tell us what skills do you believe the next generation of genomic scientists absolutely like need beyond technical expertise, in your opinion?
Okay, well, that's a great question. And I could list the expertise that I don't have that scientists coming forward need to have, right? Because I'm now an older generation of scientists. We have a set of techniques that we relied on and we used, and there's so many exciting new areas. Now that we have these huge, huge data sets, we really need machine learning to sort through those data sets, make sense of those data sets, start to make new predictions and validate those new predictions. So in my lab, I have a graduate student, Alexander Howard, who has been carrying out several machine learning experiments for the lab, and I've been learning how powerful it is. So I do think that is going to be very important for genomic scientists because there's so much more data. So when we isolated XA21, if you can imagine, there had been no genome sequenced at all. So we were just doing map-based cloning, moving very slowly, everything very simple. You find a gene, one gene, you test that gene in a plant, very methodical. And now there's so many more sophisticated ways to identify gene candidates and to validate those candidates. Although the validation has not really changed that much. We're still, in my lab, mostly when we want to validate a gene, we go into the rice plant. But I had a very talented postdoc, Ellen Rim, who's now at Stanford, that developed a new way to predict whether a gene will function using a yeast approach, so a single cell approach, so expressing variants. She could express 10 million variants in a cell very quickly. She developed a method to test if they were active in resistance, and then at that point, we would have a very small subset to test in plants. So that's also been a huge advance that we really had hoped to do even 30 years ago. And I should say, when many labs had isolated these genes, we thought, okay, now we can move them from plant to plant. We can make, hybrids, develop new methods of resistance for pathogens that we had not been able to control before. But it really did not move forward. There's maybe a couple cases, interesting cases, but very, very limited. So the approaches that Ellen developed, really able to generate and validate novel resistance genes, that I think that type of approach is going to be very useful well into the future.
Yeah, that's amazing. And we see how much you are excited about research. It's really, really beautiful and inspiring. And now I'm thinking, like, can you tell us what continues to motivate you after a decade of breakthrough research?
Well, thank you. It's definitely the people. I am so lucky to have just really a wonderful lab group. And truthfully, as you get older, as I was saying, we don't really have the techniques. So I get to see how they're creating new ideas. We have collaborations within the lab, a really supportive group helping each other. And I mean, I really, so also when you're a young scientist, you're so busy. You're trying to get grants. You're trying to figure out what your job is. You're writing all this stuff. There's so many deadlines. Some of us have kids. We're rushing around. And I have to say that, mentoring is not a priority. I mean, you want it to be, but it can't always be, because there's other priorities. You have to bring in the money, and you have to prioritize your family. But now I love mentoring. I love sitting around talking with my students and postdocs, and it's super exciting when they have job interviews and the whole group gets together and helps them. So I really appreciate that I have that time. Children are off. Funding is uncertain. We still are trying to write grants, but I'm not, as attached, really, because at some point the funding is going to end. I'm going to have to stop doing my research. So I start to see that. And so I put more time into just talking with people.
That's, I think, that's the also what I like about science, and what I like about this field. It's like the people, and it's very important. And I would like
maybe I could add one more thing.
Sure.
that international collaboration. We are so very lucky to have an international community. I really don't know any other jobs where you get to know your colleagues, your French colleagues, or your colleagues in China, Israel, and you really feel science is a global enterprise. We're all working hard together, and we want to support each other to move forward, especially in very, very difficult times when we have countries that are have poor governments, not respecting human rights, not respecting science, it's even more important that we support and help each other.
No, 100%. And thank you so much for highlighting this point and this. It's very important. Now I would like to move toward, because this is a Women in Genomic webinar. So we're gonna move to more question toward this, and I'm very curious to know, because you've been in this career for a long time, and I just wanted to know, like, if you can share a moment where you had to advocate for yourself as a woman in science.
Okay, that's interesting. Well, I can remember back, maybe even in college. In college, I wanted to be a math major. I loved math. And I jumped into an advanced math class. I think I was only 17 years old. I'd gone to college a little bit early, and I was the only girl in the class. And I didn't really think about it until later, but I knew I was very shy and uncomfortable in the class. I didn't have really students that I could go home and work the problems through with. I was aware other students were doing that. I felt very uncomfortable. And I remember going to my professor and saying, you know, I'm not going to be a math major. And looking back, I really think it's because I felt isolated, the only woman, but I didn't realize it then. And I had no idea. And I just remember he almost started crying. He really wanted me to continue. And looking back, I realized he was a huge supporter of women in science, probably a very unique person. He was a very gentle person. I wish I remembered his name. I wish I could thank him. So he advocated for me. I just wasn't mature. I didn't know what was going on. 17, I didn't advocate for myself. Let's see. I am not sure if I ever advocated for myself as a woman in science. Like I don't think I ever stepped forward and said, no, you know, I want to do this, and I'm the only woman in the room, so I need to do this. I don't think that really ever came up. Is that what you mean?
Yeah, if any moment, you know, like you had to put yourself out and say, like, I'm here as well. Like I'm not just like, yeah, because I feel like sometimes women are neglected in some research, in some like, and they're not taken seriously.
Well, I think that probably every talk I gave in my earlier years, every paper I wrote, every student I took on, I had to, you know, especially public speaking or in groups of people, I really was shy. So I had to push myself. And there were moments, I guess this is not really advocacy, but I remember I was at a meeting and it was organized by a European, but it was in the United States. And in some ways, I felt that France was a little bit behind in feminism. I lived in France for a couple of years, and this meeting was organized by a French man. And I remember the first evening, I couldn't connect with people, and I was feeling uncomfortable again. And I realized that people were like, they were taller than me. They were talking, seriously, they were talking over my head. Like I'd be there, uncomfortable standing there, and they would just be talking to each other. And then later on, a man just shoved me some plane tickets. He said, I really need you to hurry up and change these plane tickets. And he thought I was the secretary of the meeting. And I mean, there's been moments like that. I just shake my head. And then later on, I realized I was the only woman on the program. And since then, of course, there has been many advocates that we don't want to have panels, only men on panels. And I think men and women have really been advocating for that. And so meetings are a lot more fun. for a woman because it's just a little bit more natural. The men and the women are speaking to each other. I mean, at some meetings, I used to have to, I couldn't be involved in conversations. So I'd find a woman in the corner. It was usually a wife, and I'd just go talk to her because for me, it wasn't comfortable trying to push in or advocate for myself. I wanted to enjoy myself. I wanted to relax. And so I just go find the women or sometimes men that were kind of off alone, and then we would have a good time.
Mm. And is it that kind of experience that led you to share the US Davis Women in Genomic seminar, series? Like I've seen that you have shared this seminar like from 2003 to 2007. And so maybe it's that kind of experience that led you to that. And for that seminar, I was wondering like what kind of insight did you gain? And like maybe you engage in these goals like of women in science and yeah.
So I think you're talking about when I led the Women in Science series at UC Davis.
Yeah.
Is that what you mean? Oh,
Yeah,
that
yeah.
was so much fun. I mean, I definitely felt that I wanted women to feel more comfortable because I had not felt comfortable my whole career. And men were welcome too. But the scientists we would invite would talk, not only they'd have a seminar about their science, and then they'd have a seminar about their life. And I just found it so inspiring and interesting. Of course, everybody's lives are different, but at the time there was a lot of women that were so concerned about if they were scientists, maybe they couldn't have children. And I thought that was just so backwards because we have a lot of flexibility as scientists. So I loved hearing other women really make these same points. And we had some fantastic people. We had Liz Blackburn, I think before she won the Nobel Prize. We invited Carol Greider, who won the Nobel Prize with Elizabeth Blackburn, who was a high school student in Davis. So we're very proud of her. I don't know how much she associates her prize with growing up in Davis, but we're proud of her anyway. And it was just the team, I remember, that put together the seminar series was great. And it was probably one of my first leadership roles. And so I was able to feel what that was like. And I mean, that was really a tremendous time. We had some money from someplace on campus to bring people in, and they were always packed. These meetings were always packed.
Mm. No, go ahead, Charlotte. If you want
Sorry.
to.
I have so we have two more questions on this topic. And so I thank you so much for sharing this. I'm really I love it because I don't know, for me it makes me not feel alone, and maybe also like for other women or like that are listening, and I really appreciate you sharing this. And always in the same topic, I'm very curious to know like how has the landscape changed for women since you started your career? And secondly, what advice would you give to women aspiring to pursue a career in science today?
Yeah, maybe I'll start with that second question. I think what you said is exactly it. When we start to feel alone, we have to remember we're not alone, and then go talk to people, build a community or join a community or just have a best friend that's a scientist. That has been really important in my career. I have very, very close friends that are scientists, and we just talk about everything. And I have one postdoc that's in a new place. She needs to establish that community. Absolutely, she will. But moving away from a small lab where you help each other and then moving to a bigger place, you're going to feel alone, and you have to just remember this is a transition. Transitions are hard. Sometimes they take 3 months. I read someplace like career transitions take at least 3 months to start to feel comfortable. So, you don't panic in the first month because it's normal. And just make that effort to meet people. I mean, when I did a sabbatical in France, I had a friend that liked to run, and we would go running together. She'd speak French a mile a minute, and she was younger than me and faster than me. And it was so funny. I don't think she ever understood how I was always trying to keep up with her French, with her running. But it was just such a wonderful part of every day. So that, it was a little bit out. We weren't working on the same project, but we were just together talking. It makes you feel less alone. And then I think things have completely changed in science, for sure in California, hopefully in Sweden. So I also did a fellowship in Sweden for a year, and there was so much sexual harassment. It was just horrible. I don't want to go into details, but it was just horrible, and no one cared. And then at UC Berkeley, there was a lot of harassment. Again, the professors would sort of warn the students, oh, that guy, be careful when he gets drunk, he's going to come after you. And he did. And the professors did nothing. And it made for very uncomfortable department meetings because people would get drunk, the men would start harassing the women, and no one cared. And now, I know when my son went to college, he was trained in bystander actions. And I thought that bystander training was, in a way, the most important, because most men are not harassing women. And they can see when one of their colleagues is, and this training, or if you're a student, same thing. This training teaches you that it's okay to step in and how to step in and what to do, because to me, that's very important, that a peer will just stop it right away before it gets to a level where there's lawsuits and things. And I know some of the men that harass women at UC Berkeley are now huge advocates of women. I've never spoken to them about it, but it's so interesting to me. I think they feel so ashamed. And I guess more power to them. At that time, you would never get caught. You'd never lose your job. And now things are absolutely changing. I hope it's not that just I'm naive, but I don't see that at UC Davis now. People are very respectful of each other. There's a lot more women, which makes a big difference. I have a daughter. I ask her sometimes, oh, did you feel like this in that class? Or do you feel like this in the lab? And she just looked at me like I was crazy. Like, no, I don't feel that way. She's also like very tall, 5'11". So maybe that makes a difference too.
Thank you so much.
Yeah, I'm very happy that you shared that, like this kind of experience and challenges. I think it's really important to speak openly of these of this subject. Yeah.
Thank you so much.
And yeah, I think it's really important to take the time to speak about that, particularly in Women in Genomic webinar. So thank you very much. And now I would like to switch a bit for another topic and talk about like leadership and mentorship, as you mentioned at the beginning of the webinar. And so I would like to know, like, how do you balance your multiple professional responsibilities? Like, you are a researcher, a professor, an author, an advocate, and so yeah, like, how do you balance all of that? And like, could you walk us through a typical day or a typical week in your life?
Yes, please tell us your secrets, please.
Okay, well, I had no secrets when I was younger. I was just running ragged. I served as the faculty advisor to the provost. I organized all these meetings. I had young kids. I was writing grants, and no one told me, you don't have to do all that. I mean, I wanted to. It seemed exciting, but I wish I had done less, truthfully, when I was younger. And then now that I'm older, really about 10 years ago, that long ago, I was just so, I think it was after about 10 years ago, I just thought, this is too much. I'm going to burn out. And I started reading about burnout because I didn't think that would ever happen to me. And I started saying no. And after you start saying no, it still takes a couple years, maybe even 3 years before things calm down, because a lot of the yeses that you had said to committees, students, etc., these are 3-year commitments. So even if you start saying no, it takes a while before you really have more time. And I do think it's important to realize that as a scientist, hopefully you're going to have a very long career. You don't want to burn yourself out. You don't want to feel like you have to do everything and then feel anxious all the time. A lot of us feel, because I mean, it is exciting. You say yes to things because it sounds really great. I developed a system with 2 colleagues that I've known for, one of them I've known 40 years. They've always lived very far away, but we've been very close. We did, river trips and backpacking and things together. We stayed very close. And at one point, all of us were feeling this frantic feeling. And we said, okay, next time I'm tempted to say yes to this commitment, whether it's a meeting or leading a committee, I'm going to call you and you tell me if I should say no. And every time it would become apparent, like to me, when my friend was asking me, I'm like, no, you don't want to do that. That's not something that you need to do, or you want to do when the time comes, when that meeting comes and you have to do this international travel. You're going to be too busy to do it. So just say no now. And so we really helped each other. And so now, as I said, I am doing a lot less. I mean, maybe then you're not the most popular person in your department, but you're helping your mentees. If you have a lot of mentees, I think they should be a priority over department commitments. Mm-hmm. And that's a little bit radical, I think, because we always feel it's wonderful to contribute to the department. It's important to contribute to the department, but you have to realize what your own capacity is. If you're feeling anxious all the time, you're doing too much.
I agree. I had that when I was doing my PhD. So I had a burnout, and it was like early. So I was like, okay, I need to just to take care of myself, because if I don't take care of myself, I won't do my PhD well. So I need to prioritize my mental health first, and after I'll do everything better, because it's very important what you're saying. And I'm very happy that I figure out this in my during my PhD. So
you can't please everyone. You absolutely cannot please everyone.
It's true, and it's hard to recognize that, and I'm really glad that you're saying that. And I was curious, because that's one of the skill that I think is very important when you're a scientist, to say no and to take care of your mental health. But another skill that I think is very important is communication. And I feel that communication and communicating in a way to make it understandable for people, it's a skill that it's very important. And because you did a TED Talk, and you reach millions of people with your TED Talk, your amazing TED Talk. And I feel like you are very good in science communicating, also with your book. And so what do you think scientists still underestimate about science communication? Why they're why they don't put all their, because it's very important communicating the science in a way that people can understand.
Yeah, I think it's not for everyone, for sure. But those of us that really love communicating science or engaging with the public, it's really just a matter of keeping things fairly simple without going into too much technical detail where you lose people, and to practice. After that TED Talk, I practiced so many times, and they actually had, what was really fun, they had experts in Israel, communication experts that you would practice with. And they were really fun and funny. And and helped a lot. So practicing, I think we all hate practicing talks. I mean, I do. It's just, hearing yourself drone on, but it is very, very important to practice. And then the book. So for those of you that don't know, I wrote a book with my husband about organic farming, genetics, and the future of food. And with my husband and we had a writing group, and they would say, even to my husband, well, what does that mean? Cultivate, you need to explain, that's getting rid of the weeds. So we had to even, for farming technical stuff, and of course, science technical stuff, our friends who were not scientists or farmers would push us, like, explain that more. And then what meant so much to me is my father, who was a refugee, never had, I think, a speck of science education. I don't think he wouldn't know what the difference between a cell and a protein, or really knew nothing. He read the book twice, and he made comments and he asked questions, and that was so helpful because I wanted him to be able to read the book. So he was someone I was trying to reach, because of course he was interested. He wanted to know what we did. He was a big reader. So I think engaging with people that aren't scientists early and practicing is important.
Yeah, thank you so much.
And since you mentioned the book that you co-author, maybe you can also talk about your vision of the future of agriculture, and maybe let's speak about what you think are the necessary breakthroughs that we need in plant breeding and in agriculture, like to ensure the food security.
Okay, well, that's a big question. I should tell you that 2 years ago, I had some colleagues that translated the book into French.
Mm-hmm.
La Table de Demain, I think it's called. And it's the best version because my colleagues that edited it improved it. So I want more French people to read that book.
You can promote
buy
it.
I can tell you where to get it when we have a chance. Okay, so what we talked about in the book, really, to us, it was very basic, but I think to the public, as scientists and farmers, it was totally basic, but the public, I think, hadn't heard it in that way before. We were talking about taking the best approaches of farming, and organic farming has a lot of really useful approaches, and bringing in the best kinds of seed, which often we made the point are genetically engineered or now genome edited. Europe does not allow genetically engineered food to be grown in France. And so that's something that I don't think is going to change in Europe, but Europe is, I think the European Parliament did recommend that genome editing be allowed. And that's a huge breakthrough. It hasn't been passed into law, but if that happens, I think it's going to open the public's eye to what genome editing is. Maybe people that are interested will think about it. Probably no one, consumers really won't think about it that much. They'll just accept it because if it's allowed, they're trusting the regulatory agencies. But that is very exciting. It still will not be allowed in organic produce. So organic, I think, is going to remain sort of a niche because they won't be able to take advantage of new innovations. It's going to sort of remain, unfortunately, a little bit stagnant. But it's very important to many people to be able to have the option to buy that niche product and But I think the rest of agriculture around the world, 99% of agriculture that's not organic, is going to move towards really reducing pesticide use. Hopefully, there's a lot of work in nitrogen use efficiency. Hopefully, we will have breakthroughs in that field so we can reduce the amount of fertilizers. That's going to be very important, as will be approaches to develop using phosphorus more efficiently. So there's a lot of genetic approaches, and then farming approaches are going to be absolutely important. Developing and less developed countries, intercropping, developing ways to boost the yield without harming the soil. A postdoc in my lab, Flora Urkely, who's now assistant professor at UC Riverside, she is working very hard to understand the microbial interactions with rice roots and thinking of ways we can engineer the plants so we can attract those microbes that are going to benefit the plants, whether it's to reduce methane emissions in rice or enhance nitrogen use efficiency. I think these are really exciting areas. Long term, it's going to take quite a while for the community to make progress, practical applications in this area, but it's certainly moving forward.
Thank you so much. Thank you so much for everything. And we have to conclude our conversation. And so we have a last question. And would you like to, what message would you like to leave for the next generation of scientists working to create a more sustainable food system?
Well, keep at it. Find mentors who are working on projects you're excited about. And if you're excited about your project, stay focused, just do it, and know that you are contributing to food security. We need a huge community of scientists working on this, and reach out to people that are happy to talk to you. You can call me up on the phone. I'd be glad to talk to you, and find community if you're going through difficult times. And remember, life is up and down. It's not only science. And when you're down, remember there's going to be another up period. So keep up the good work.
Thank you so much.
Yeah, thank you very much. Very good advice. So now I think we are a bit almost out of the time. So if there are any additional questions from the audience, you can enter the question in the Q&A box. And yeah, Deb will try to get them. So
yeah, thanks, Laura. Thanks, Laura, and thanks, Charlotte. I think you did a great job with the conversation with Pam. And thanks, Pam, for sharing so much great advice. I mean, I learned a lot. I do want to pull up just a couple of questions from the Q&A chat box. One is from Jan Leach. You mentioned Jan is having a role in your training, so glad to hear that she's on and asking a question. But she says that, your lab has had a long history of successful collaborations with international scientists and labs. And so what advice would you share with young scientists wanting to build programs with international collaborations?
Well, first, of course, I have to say thank you, Jan, for joining in, and more kudos to Jan. I only mentioned her in a passing, but she has been tremendously important in my career, not only answering a phone from a graduate student that knew nothing about Xanthomonas oryzae and helping me, but throughout my career, very open, ready to talk, ready to discuss. Completely unpretentious. Jan is a wonderful person, and I still value her support very much. And look at her, she's showing up to this talk. So thank you, Jan. And young scientists wanting to build international collaborations, for me, it has always been the students. So having a student for me, it was China, because I worked on rice. And at the time, the United States had this great opening with China. There was a lot of scientific collaboration, a lot of support. I never dreamed that we'd be in a situation like this now. But those students, they'd come over with the Chinese Scholarship Council supported by the Chinese Scholarship Council, which has now been disbanded because of the problems between the countries. But they would come, work so hard, teach me a lot about what their labs were doing back in China. And many of them have gone on to establish their own labs, and we've continued to collaborate. So I think international collaborations do often start with the students. Going to international meetings, also, if you meet somebody that wants to work with you, that also can be a very exciting type of collaboration. It can go forward, but what I find sometimes the principal investigators have these great ideas and they want to do it, but they go back to their lab and no one in their lab is excited to do it, or in the other lab. So you need to have a person in each lab committed to the project, or it won't move forward. So thanks for that question.
Yeah, that's great advice. And sort of a related or follow-up to that, you talked about the importance of having students that are excited about projects and wanting to work on these new ideas. So can you share your approach for guiding trainees in genomic science? Some people feel like they often benefit from more structure as they learn genomics, and working through projects sometimes can feel very daunting, right? Overwhelming if you don't know the technology or the science. So any tips to help beginners, especially those that might have different learning styles?
I think it's important for a graduate student to have really work closely with the postdoc at first to learn the techniques. I mean, it's like a big kitchen. Where's everything stored? How do you work this machine? I think that is one aspect of our field that most of us do, which I think is fantastic. My son's an ecologist, and I know from my friends that are ecologists, it's really sink or swim. You start your own project, you have no mentors, you got to figure out how to use the machine yourself. I think our approach is better, where we can pair people up, and students will feel very supported. And then when they start developing their own ideas, we have lots of conversations, and then they go off on their own. So for me, that's the way I've run my lab, and I feel like it helps support students quite a bit.
Great, thanks. And then there was one question about the gene validation. Early in your talk, you talked about validating genes. Has that method been published, or can you discuss how that gene validation method worked, just in brief details?
Sure. I mean, it's simple, but for example, sub1, my postdoc in my lab identified he mapped a very small region where the sub1 locus was, in collaboration with Dave McKill, who's a fantastic geneticist. And then he created a physical map back in the days when we didn't have sequence. We finally just threw away our back libraries. And then he had a bunch of very large clones that he thought sub1 was in the region, but he transformed them all into rice. So in this case, validation was taking those plants, many, many hundreds of plants, and growing them for 2 weeks, submerging them for 2 weeks, and seeing which ones would survive the submersion. So validation is simply, you can have, even in the future, when we're having great computational predictions, you always need to put that gene in the plant, in the field, to see if it has the effect that you hypothesize. So essentially in my lab, validation means getting that gene in the plant and testing it to see if it confers the phenotype that you're looking for.
Yeah, great. So it's really just getting back to the physiology or the function of the plant, with or without those genes, right? In the growing situation. So, well, that's all the questions from the chat box or the Q&A box. So thanks again. You shared so much important information: science, your career journey, your philosophy on teaching and on training and research. Two things that I really appreciated was the the advice you gave about finding a community, right? Building your community. It does, and I think that's one of the whole purposes of this group, Women in Genomics, is just to learn from people that have, been through some of the challenges or how they've thrived in their career. It's not always about science. I think you talked about find a running partner if you're into running, or go have coffee with somebody, or a food. Food brings people together from all cultures, right? And I think just talk through some things, talk about your challenges, talk about what's good or what you need help with. And I think community is just such a powerful mentoring tool or support group. So I really like that idea. And then work-life balance. How many times have we said, oh, just say no. You don't have to do everything. You don't have to do it perfectly. Just say no. But in my experience, it's really hard to do because it's so fun. I like doing things, and I can, I've been around long enough. I know how to get things done or who to talk to. But as Charlotte said, we really need to be aware of our own mental health and take care of ourselves when it comes right down to it. And I think the holiday season that we're all in right now sort of brings that home too, right? We're all trying to get things done before the end of the semester and get cards out and presents wrapped and all of that. But we just need to protect ourselves and take care of ourselves. So this is great. I just want to thank you again, Pam. This has been fabulous. I look forward to seeing you at the LGBT Ag meeting in Phoenix and your talk. We
Yeah.
put a bunch of those links in the chat. So I hope who's ever participated in this webinar will click on those links and find out more about this group, Women in Genomics, as well as the workshop and the dinner at the Plant and Animal Genome meetings in San Diego in January, and then the LGBT Ag meeting in Phoenix in April. So, and there is a discounted registration for this group. If you do want to go, and if you're a little tight on funds, there is a discounted registration, and that code is in the chat as well. So I think that's it, everybody. We did really, really appreciate the conversation. And for those of you that do participate in the quarterly virtual meetings, we do plan to organize one for the end of January. After the plant and animal genome meetings, we'll kind of have a debrief on that meeting. We do have a panel of women and male allies to talk about women in genomics at the plant and animal genome meetings as well. So anybody else have any other parting comments?
Thanks again, Pam. It was really, really cool, this conversation, very insightful. So thanks a lot. And yeah, thanks to the audience.
Yes, thank you so much for your honesty and being humble and everything, all your insight. It was awesome to have you and to get to know you more. So thank you so much.
Well, thank you all. Great questions. And I also look forward to meeting you in person. It'll be wonderful.
Yeah, I can't wait.
Okay. Thanks, everybody. See you
Thank
next time.
you.
Thank you.